Hi, I'm Anthony Originello, CEO of the Chartered Professionals in Human Resources for BC and Yukon. We represent more than 8,000 members, and as the organization dedicated to advancing the HR profession through education, advocacy, awareness, professional development, as well as our recognized and impactful CPHR designation, we are proud to be a sponsor of this show. We have been the voice of the HR profession since 1942. Learn more about us at cphrbc.ca.
On this edition of Careers That Matter, I'm joined by Dr. Christopher Mason, who is with Weill Cornell University, and you've got a variety of different disciplines, one of them being genomics. Tell us the rest of what your responsibilities are within the university, and then I want to talk about your career.
Sure. So I'm a geneticist and a professor there, which means we have a laboratory that does a lot of work in genetics, which includes looking at the DNA of different species, including humans and their microbes. We do also a lot of computational biology. Half of our laboratory is a dry lab, meaning it's just people working at computers, because so much of the data is complicated and requires AI tools and algorithms to process the data. And the third part of the lab is really looking at synthetic biology. So can you combine different pieces of creatures that, like a human cell with a tardigrade, or mix and match different strains of bacteria to make it so plants could grow on the moon? So it's a way to mix different components of life in new ways. So synthetic biology means you're creating new kind of biology. But it's a laboratory, so there's people that work in the lab. You can imagine people at the bench and doing experiments. But also we do a little bit of teaching for the medical students and the grad students in the spring. But really, about 95% of my time is in the lab doing research.
So how many people in the lab sort of fall under your area of responsibility?
There's about 30 people, which includes graduate students, people getting their PhD, master's students, undergraduates, medical students, and postdoctoral scholars, people who have a PhD, but also are then following up with different research. So it's a really wonderful and eclectic group in the lab. Very honored.
So in essence, you're running a small company.
Yeah, basically, because we have to find money, we have to write grants, we have to get money to pay salaries, run experiments. It's a small business. It's the best analogy. Yeah.
Yeah. Well, that bit about applying for grants, that's a fair bit of work too.
Yep. Yeah. I'd say even 20% of my time is either writing a grant, updating a grant, responding to some aspect of an update related to a grant. So there's a lot of upkeep just to make sure we keep the funding in place. But that's true for a lot of small businesses as well.
Well, you mentioned also that you do some teaching. Mm-hmm. How much of the overall year is, I don't want to say consumed, but dedicated to teaching?
The teaching is about 5 or 10% of the time. It depends on the year. Some is a little bit more, a little less, depending on which classes are active that year. But so it's the minority time, but it is really a great honor to get people excited about the latest technologies in genetics. What are the new algorithms? What are the new opportunities in science? So I really enjoy getting the next generation of scientists up to speed with what's the latest and greatest tools, methods, algorithms, but then also get them excited about the creativity side of science, where they can imagine an experiment or design a new organism quite literally, or design new concepts they could then try out in lab.
So, do you work 24/7?
It is in a way, yes, I suppose. But if it does sound cliche. But if you do what you love, you never work a day in your life. And so I very, very much love the lab, the experiments, the data sets. There is some paperwork, which is not as exciting. I have to fill some paperwork out, so that's not the exciting part of any job. But almost every day, if I could, I'd be working on questions in science and genetics and lab. Yeah.
So when you wake up in that space between actually being awake, that delta sort of state, is your mind just like at work going, what about this, what about that, how about here, how about there?
What about this experiment? Yeah, writing little notes down all the time of what if we try this other data set or different experiment all the time. Yeah, you can ask my wife. She's like, why are you always writing ideas down for experiments? But it's an unprecedented time. There's never been more data than ever before in science, especially in genetics. And so that means every day that I wake up, there's more data than the day before. So that means quantifiably, every day I wake up is the best day to do my job, and that will always be true.
Okay, I got to ask you this just because I think it's silly and but fun. But Isaac Asimov said, great breakthroughs don't come in some eureka moment. They usually happen when you look at something and go, well, that's kind of odd.
Yeah.
And is he right?
Yeah, I'd say those are often some of my favorite times. There are times when you do see an unusual result, and you know that it's exciting and it's surprising. One example would be when we first looked at the number of genes that are present in our DNA. So you think of the human genome as DNA, and there's a certain number of genes that make hair or eyes or enzymes or your heart muscles. And originally the thought was that the human genome only had about 20,000 genes, maybe 25. But it turned out it probably has about 50 or 60,000. So when we first got some of that data back in our lab when I was a PhD student, that was kind of a eureka moment because we knew there was so much more than what we thought before in the cells we were looking at. But there are just a lot of times where you look at something and it's like, why is that there? And what does that mean? And it just seems strange. And that is probably the more common clue towards a new biology or a new discovery.
Yeah, there's something worth exploring
Yeah,
there.
yeah.
So that's your work at Weill Cornell, but you're also working with NASA and this twin study. How much of that consumes your time?
Well, the big study had finished in about 2020, so we finished a lot of the analysis and the data collection. But it's ongoing because we want to look at long-term follow-up for the astronauts as well as other ones we've looked at with SpaceX and some Axiom crew members. So there's now these commercial spaceflight entities like SpaceX, Axiom, who are flying their own crews. And so we've been working on some of those missions to see, well, we follow them during the mission, but also long-term follow-up. So it takes only a bit of time, but it is building really a lifetime resource for the crews to see how their long-term health is, if they have any long-term risks, which you can only see if you follow someone for 5, 10, or 20 years.
Okay, so let's take a look at the course of your career. Where'd you grow up?
Well, originally from Wisconsin, Racine, Wisconsin, born and raised, and then went to undergraduate in Madison, Wisconsin. It was one of the few schools you could get an undergraduate degree in genetics, which I knew I wanted to do genetics ever since I was an 8th grader when I learned about embryos. And so,
why? What was it that made you go, okay, I want to do that?
Well, I'd known about embryos before, but we've gotten into the genetics of embryology in my 8th grade science class, and also the chemistry of how it works. And I thought it was extraordinary that you have a single cell that has all the instructions to make every other cell in the body, and is the seat of how you get cognition, what is eventually going to become your dreams, your aspirations. It is all present in that first cell, and we just need to understand how that static code manifests into trillions of cells and how it can then respond to and eventually age and eventually die. But that all the clues are already in that genetic code from the very first cell. So
by the time that you had graduated from high school, you knew the direction that you wanted to pursue.
Absolutely. It was genetics. If anything, I was very frustrated in my undergraduate years because you don't start taking your more dedicated classes till junior year, senior year, or later. And I really wanted to get going at genetics. Like I knew exactly what I wanted to do. I don't have to take this basic chemistry class, take some other physics, but I really wanted to just do genetics. So I knew that right away.
So what degrees do you have? I mean, you're Dr. Christopher Mason, but what specifically did you study to wind up with your PhD?
So studied a lot of which genes are present in fruit flies and in human cells. So that was I got my PhD in genetics from Yale. So in a lot of times you finish your bachelor's degree, you can either go get a master's or get a PhD or a medical degree. And I knew I wanted to do the research in particular, so that's why it's good to get a PhD for the scientific training. And I knew I wanted to do genetics, so I applied to the best programs. I just downloaded the list of the best genetics PhD programs and applied to the top 5, and that was what I went for. So, and Yale was one of the best ones. It was number 2, so I got in. I liked the people. It was great teams, great labs, and so I joined there in 2001, and then finished my PhD in 2006. So.
And where did you transition from there to actually getting paid for doing the work that you're doing?
Right. Well, one good thing about grad school is you do get a stipend. So actually I was getting, that was the other thing I was surprised. I was like, wait, someone will pay me to go get my PhD? Like you get a stipend to live on. You're not going to be wealthy, but you can survive and pay rent and just if you want to work in lab, it is great. So so it started that in 2001, but then also did postdoctoral fellowship training at the Yale Medical School to look a lot at how diseases are inherited in different families. And then also a lot of the new technologies to look at genetics. And then that was till 2009, and then started my laboratory at Cornell.
Wow, what a ride.
Yeah, and it's been extraordinary because the beginning stages of my PhD and postdoc with some of the new technologies were just emerging that let us look at large scale, all the changes across our genome and DNA of any species. And they have all accelerated extremely fast. So the ideas for an experiment I had 20 years ago that seemed impossible, we can now do routinely in the lab today. Like if you want to reach into a cell and control genetics, we can do that today, but that was pure fantasy even 10 years ago.
So for a young person who might be watching this going, wow, I want to be you, what would you say would be a characteristic that you've learned about yourself that you have that was essential to your success?
I think a few traits that I think have been helpful. I would argue in success, there's always going to be someone better than you. There's always going to be someone a little bit worse. The race is long, and at the end, it's just with yourself. So one thing you can't do is always compare yourself to someone else, because there's always someone better, there's always someone worse. But so that's the external factors, and I always try to focus on just what are the reasons I got into genetics. So just be curious about how life is and how it could be. So having something that you're curious about and passionate about was key. Being dogged and dedicated and persistent, because most people, I've had grants get rejected, papers get rejected, experiments fail. Failure is part of science, and actually, if you do an experiment well, it is the best teacher in science. Is this failed, but why did it fail? What did I not understand about a cell or a system, or even a grant? You apply for a grant, or you submit a paper to get published, or apply for any fellowship, or even you will not succeed all the time, and that is probably about the almost only thing you can guarantee, except for death, taxes, and failure. You will not succeed every time you try, and that is part of living, and to not take it, as the end of the world if something didn't get funded or you didn't succeed or an experiment failed. That is to be comfortable with failure, and even to expect it and to cherish it.
When something goes sideways for me, as I run a small business here, I go, well, you never know when you're having good luck. Because I find that when something goes sideways, it forces me to look to a place that I might not have otherwise looked at.
So you have to be adaptive and really persistent. Yeah, definitely. So, but the biggest thing is, if you really love what you're looking at and are curious about what you're working on, it will never feel like it's a work in a normal way. It's just something you would do. If it's something you would do for free anyway, and you love it and you're curious about it, then it's just a bonus to happen to get paid for it as well. So to find the thing that you're most curious about and do that.
Well, thanks for sharing insights into your work and your career, but also into your philosophy on life.
Yeah, my pleasure. Well, thanks for having me.